today we are going to talk about some very important topic from human reproduction from your NCERT and which is very less explained. So, we will try to understand in easiest manner, right. So, the name of topic is gastrulation. What is gastrulation? Gastrulation, as you know, gastrula arises from blastula, right. In blastula there is the presence of inner cell mass. So, gastrulation is the process of morphogenetic movements of inner cell mass to produce three germinal layers, that is ectoderm, mesoderm and endoderm, and this process is termed as gastrulation. So, before starting with the actual process that takes place during gastrulation, we will understand some features of gastrulation. So, gastrulation, first feature is gastrulation is very complex process. Though it is complex, it is orderly and irreversible change that is going to take place in inner cell mass, right. Second thing, cellular division rhythm of cellular division goes down. Third event, nucleus become third feature nucleus become more active. Fourth one is type of metabolism changes in this case and the rate of oxidation is intensified, and lastly chemodifferentiation starts or it takes place by synthesis of various types of proteins. So, all these are the features of your gastrulation in case of animal. Now, there are some formative movements which take place during gastrulation, and there are basically two formative movements. First movement is epiboly and second is emboly. In case of epiboly, there is a stretching and spreading of ectoderm forming cells to form ectoderm which surrounds embryo except at one point, and that is blastopore. That means, keeping aside blastopore, entire embryo is going to be surrounded by cells of inner cell mass or cells of ectoderm forming cells. Second thing, emboly. What is emboly? Emboly is it is the invagination or spreading of cells of inner cell mass, so that internal layers of your mesoderm and endoderm would be produced. So, these are two formative movements take place during gastrulation. Emboly again can be divided into various methods, that is invagination, delamination, ingression, involution and convergence. These are different methods of emboly. So, we will look into the first diagram. Now, we know that zygote is formed in your ampullar region of fallopian tube at the junction, that is ampullary isthmatic junction. Now, it migrates and it reaches uterus on third day and it gets implanted in the form of blastula on 7th or 8th day of fertilization. So, consider this is your implanted blastula. This outermost layer is of endometrium and this layer it is called as trophoectoderm, or simply it is a trophoblast, right. Now, these cells which are gathered at some point, this end is called as embryonal knob and the cells are of inner cell mass. The cavity produced is called as blastocell and this blastocell is filled with some fluid which nourishes developing embryo in the initial stages. Now, we can see these are red color cells I am drawing here. These cells are which are in contact with inner cell mass. These cells are termed as cells of Rauber, and trophoblast loses its boundaries and it invaginates or it enters into the endometrial linings, and this trophoblast now is termed as syncytiotrophoblast. Well, now this is the implanted blastula, clear? Well, in the next stage, this is the second step of gastrulation, we can say. Now, here is the implanted blastula. Now, cells of Rauber in this case, in the second stage starts degenerating. Here we can see the cells of Rauber degenerates or it undergo degeneration, and inner cell mass cell arrange themselves into some structure or some disc like structure, and that is called as embryonic disc, right. So, in the next event what happens, as the cells of Rauber has already been degenerated, this position or this location is taken place by your embryonic disc. Now, this embryonic disc moves to the place where there was presence of Rauber cells earlier, right. Now, this is our again blastocell. This is trophoblast. In the next one, what happens? The cells of embryonic disc undergo division. It divides and it forms two layers. There is columnar and lower one is your cuboidal layer. Upper layer is called as epiblast and lower layer is termed as hypoblast. Now we need to understand in this case is columnar cell, that is epiblast layer, is going to form embryo in future. This hypoblast do not participate in embryo formation, right? Well, so what the next event, this is your third event, this is fourth, this is fifth event. Now we understand the cells of embryonic disc has undergone division and it formed epiblast, hypoblast. Epiblast forms embryo, hypoblast it do not take part in formation of embryo, right? In the next event, cells of this embryonic disc, that is a epiblast, some cells delaminates from this layer, from embryonic disc or this columnar cells or epiblast. Some cells delaminates and it proliferates and in a circular manner to form some structure, and that is called as your primitive gut. The cavity is called as actually primitive gut, or it is also termed as archenteron, and this is the formation of your endoderm. Endoderm is the first layer to be formed during the process of gastrulation, right? So we will understand this once again. Now this was the embryonic disc, that is epiblast layer. Some cells from this layer delaminated, it circularized and it formed endoderm, and the cavity of which is termed as primitive gut or archenteron. Next layer to be formed is your mesoderm. Now what happens? This is the embryonic disc. Now this end is termed as cephalic end and this end is called as caudal end. Now cells from caudal end start proliferating and number of these cells increases, and now these cells will detach from this proliferated part and it will scattered into the blastocele to form some patches, and these patches will give rise to your mesoderm, because mesoderm in future after morphogenetic movement it is going to form some tissues and organs, right? So second layer that was formed is your mesoderm layer. Now after the formation of mesoderm, the last germinal layer to be formed is ectoderm. Now remaining cells of this epiblast, it differentiate I mean it scatters in such a way that it lines up the internal boundaries of trophoblast, and that forms your ectoderm. This is how three germinal layers are produced, and now we will see some how amniotic cavity is produced, because it is related to this, right? So as already we had said that cells of robber underwent degeneration, so it kept it left some cavity here, and this cavity which is going to increase in size and it will surround developing embryo in future, and this cavity is termed as amniotic cavity, which will be filled with amniotic fluid. And we know the use of amniotic fluid, that is for amniocentesis, to identify various disorders, genetic disorders which may take place in a growing fetus, or to find out the sex of that fetus. Hope that you have understood this process of gastrulation in the easiest way. So in the next video we will see how extra embryonic layers are formed. Thank you.